Hello, guys. Welcome to the knowledge domain, the concept of life sciences and biotechnology. In this lecture, we are going to learn the mapping of chromosomes. The chromosome maps are called the ideograms. Genetic map provide the information about the location of genes in the genome that provide the ability to identify and isolate genes. W. Sutton in 1903 first time provided the idea that the gene resides on chromosomes, or genes are located on chromosomes, or genes are found on the chromosomes. Then in 1910, T.H. Morgan experimentally worked that developed the techniques for the gene mapping. And in 1911, the world's first chromosome map of the Drosophila was prepared by the Stuart Wendt. After 11 years, in 1922, comprehensive analysis of the relative positions of over 2,000 genes on the 4 chromosomes of the Drosophila melanogaster was prepared. Then in the 1940s, Avery, MacLeod, and McCarthy in 1944, Hershey and Chase in 1952, given the concept that DNA is the genetic material. And then we all know in 1953, the double helical DNA structure was proposed by the Watson, Crick, Wilkins, and the Franklin. The goal of the chromosome mapping is to construct correlated genetic and the physical maps with the markers distributed at relatively short intervals throughout the genome. There are 3 types of the map, and we'll discuss each of them. First, we'll provide a summary of all the 3 maps on this slide. Genetic map is also called the recombination map, or it is prepared by the recombination frequency between the markers. 1 centimorgan equivalent to distance that yields an average recombination frequency of the 1%. The molecular markers in the genetic map can be an RFLP that we will discuss in one of the following slides. RFLP stands for restriction fragment-length polymorphism. Then the second type of the map, the cytological map, or that is prepared by different type of the dye or the stain by preparing the bending patterns of the various chromosomes. And each of the chromosomes can be identified based on the shape, size, and the length, so that we can identify and we can name, we can give the different names to the different chromosomes. So location of markers within near the cytological features, because the chromosomes in the cytological map can be having a number of the chromosomes, then it has the 2 arms around the centromere. One is the long and other is the short, that we will see on the image. Then the third one is the physical map. So the restriction map, this is also called the restriction map because we can use the restriction endonuclease in preparing the physical map. But physical map is the sequencing of the chromosomes. So we can represent it by the restriction mapping or by the base pairs, kilo base pairs, or the mega base pairs, the million base pairs. So high density map can have all the 3 features: genetic, cytological, and the physical mapping. And we will discuss the Contig in one of the following slides. So this is the representation of the 3 types of the map. This is the genetic map or the recombination map. And the units is expressed as centimorgan. So 1 centimorgan equal to 1% of the recombination frequency within the 2 different genes. And we can see the molecular markers. One of the examples is the restriction fragment-length polymorphism that we can be in the recombination or the cytological map. This is the cytological map. So this is prepared based on the bending patterns using the different types of the dye. So based on the shape, size, and the length of the chromosome, we can give the name in number to a chromosomes. After that, the one chromosome is having a long arm and a short arm. So the short arm is the P arm and this long arm is the Q arm. Then this is the centromere positions. And after the centromere, we can name the number of the band. Similarly, we can locate the position of a gene based on the first we'll give the chromosome number, then it is the long or the short arm, then which is the number of the band. Then after the number of the band, we can give, it can be with the decimal also. There can be another number after a decimal to give the smaller bands in the structure of the cytological map. Then we have the physical map that is completely based on the sequence. Then the restriction mapping that we will discuss in one of the following slides. So one small portion, these three are interconvertible that also we will discuss. So we can give the positions to the different restriction endonuclease based on the how many sites it has and their sequence or the order of the restriction endonuclease in the chromosomes. So this is called the restriction mapping. Then Contig is the contiguous mapping. So here we can prepare the smaller overlapping sequence of the DNA by taking a special location of the DNA. We will clone them, we will amplify them. After that, we will sequence each of the contig. And after that, we will overlap and we will prepare the sequence of this entire area. Then this is the STS, the sequence text site. This is also a molecular marker. So how they are interconvertible? This is the genetic map prepared based on the recombination frequency. Then each of the locus or the gene can be placed on a specific bands of a cytological map, and each of these positions can again be traced back to actually the nucleotide sequence of a physical map. And finally, this is the DNA sequence of a chromosome. So genetic map, cytological or the cytogenetic map and the physical map, they can be comparable to each other, they can be interconvertible to each other, and we will get a high density map or a accurate information by having holistic view of all the three types of the map. So how they are interconvertible? So there are the clone genes can be positioned on the cytological map as we have seen in the earlier slide. The anchor markers or the molecular markers can be present in the genetic as well as in the physical map. Then there is a technical, the radiation hybrid mapping. So here the smaller fragments of the chromosomes, the DNA are prepared by the radiations so that it is a fixed amount of the radiation that is expressed in the gray for a specific period of time in a specific situation. So we usually get the similar fragments. Those are the fragments, usually very reproducible. That's why it is very much used. Break the DNA into the fragments. The amount of the radiation can be adjusted as I discussed. Then the genetic and the cytological map. So it is a PCR based 200 to 500 base pair because in one reaction we can sequence up to the 800 nucleotides. Then the specific place can be identified using the probes, and those probes are used by the southern blot to relate the sequence to overlapping clones on the physical map. Then the in situ hybridization to determine the chromosomal locations in the cytological map positions. And there the sequence text sites are the short, unique molecular markers or the anchor sequences, so that it is a type of a landmark in case of the geographical map, so that we know RFLP, we know the STS, we know the EST, and around that area we can identify or locate the place of the genes or the place of the sequence of the DNA. Then there are the short cDNA sequences, the complementary DNA sequences. Then the express sequence tags are the PCR based, and here the putative genes, the functions yet to be assigned for the putative. So we know the location of a gene, but we don't know the frequency, the function of that gene. That can also be used as a positional cloning or the positional mapping or the molecular markers. Then there is the exon trapping method or the exon amplification may be used to identify the exon sequence. Then the hybridization probe to anchor the physical map. so that we can give specific molecular marker position to a specific place in a cytogenetic map. We can compare it to the genetic map and finally to the physical map. As we have seen in the previous slide, RFLP is a molecular marker and it gives a identifying feature at a specific location of the chromosomes. So recombination frequency are yet always proportional to the molecular. It is not always proportional to the molecular distance. So we can convert or correlate genetic to cytological and genetic to the physical map. So what is a genetic map? In the genetic map, we get the information about the linkage, means how the two genes are related. So genes on the same chromosome should be inherited together. Based on that, we have the concept of the linkage, and they travel as a unit through the meiosis. Then physical, we have the concept that two genes are physically attached to the same structure, and linkage is not absolute. So crossing over, we can see it through the chiasmata, where the exchange between the paired chromosomes takes place. Recombination, the separations of the linked genes. Formation of the new gene combinations, result of the physical events of the crossing overs. This all happens because of the recombinations. Recombination frequency measure the intensity of linkage between the genes. Genes that are tightly linked seldom recombine. Genes that are loosely linked, they recombine very often. So if they are recombined very often, the recombination frequency is very high, so they are far apart. If they are tightly linked, the recombination frequency is very small and they are very close to each other. For any two genes, the recombination frequency never exceeds 50%. So if between the two any genes, the recombination frequency exceeds 50%, means they are very far apart or they are in the different chromosomes. The total frequency of recombinant progeny from a test cross involve two genes on the different chromosomes will be 50%. Then a map unit, we have discussed in the previous one, 1 centimorgan is equal to almost, in physical map, almost 1 million bases DNA. So map distance is equal to the frequency of recombination written as the percentage. Then the restriction mapping. After getting a holistic view of the physical cytogenetic map and the recombination map or the genetic map, we will understand how the maps are prepared. So restriction endonuclease are very important tools in preparing the map. So how to prepare a restriction analysis that we have seen in one of the previous slides. So we need to know the relative positions of the restriction endonuclease. So correct restriction endonuclease be selected for the particular protein manipulation. Then we need a series of restriction digestion. The number and size of the fragments can be known by the gel electrophoresis. Then we compare with the size markers because we have the molecular markers has to be run in the gel. Then we need a series of double digestions. One step if both enzymes have similar requirements like the pH, magnesium concentration. Two digestion may have to be carried out sequentially, adjusting the reaction mixtures after the first digestion to provide a different set of conditions for the second enzyme. Then there is a partial digestion because you don't need it to fully digest everything. So the cleavage limited number of the restriction sites on any DNA molecules. In practice, more than 50 kb cannot be resolved efficiently using the standard gel electrophoresis. So this is an example of the restriction digestion or the restriction mapping. So in no digestion, it is the 9 kb with enzyme A. We can see there is the 2 fragments, one is the 2, another is the 7. With the enzyme B, there is also 2 fragments, one is the 3, another is the 6. Okay, so it can be this way, it can be the another way. Then when we combine the two enzymes A plus B, it is a partial digestion, so we can get 2 and 3 plus 4. So finally, or alternatively, we can get that 2 plus 6. Okay, so how it is possible that in the enzyme A, there it is the 9 kb total. So in enzyme is 2. Okay, so enzyme may be present in the 6 fragment. So this 2 will remain as intact because when both enzymes are present, one is the 2. So another one can be in the beginning or it can be in the end. So this is the 7. So the 7 is divided into the 2. 3 plus 4. Okay. So this can be on this side the 4 or it can be on the other side the 4. So in the second, the B enzyme can be found here or it can be found very close. So similarly, when we have more than the 2 restriction enzymes, it will be slightly more exhaustive. It needs some more work to identify the relative positions of the different restriction endonuclease. So in these cases, the positions of the A and B can be either this one or it can be either one. So we can see here the 2, okay, with an enzyme A, and in this case, the B is present between the 4 and the 3, or it is between the 6 and 1. So in both cases, we can have this 7 kb length. So next, the same restriction digestion can be used for the RFLP. So in this case, this is a special location in this one. So we have the 3 equivalent restriction sites. So there is the G8, etc, G8, etc, G8, etc. So here we can see there are the 3 bands. So one is between this and this one, and another is between this and this one. So in the DNA ecotype 2, there is a mutation here. So instead of GA, it is GGA. So there is equivalent restriction site is removed from here. So in the ecotype 1, we can see there are 2 bands. Here in this case, we can see the 1 bands. So for the different ecotype, there are the different RFLPs. So how do we prepare it? We isolate the DNA, we digest with the EcoR1, then separate it, then transfer to the nylon membrane for a southern blot, and after that we hybridize it and we do the autoradiography to see the different bands. So in this case, we see the 2 bands, ecotype 1 in the second one because there is a mutation, so EcoR1 cannot digest it. So instead of 2, we can see 1 bigger band here. So the RFLP help us or allow us in differentiating the 2 types of the RFLP that is possible because they're from the 2 ecotypes. So what is an ecotype? DNA of the different geographical isolates are called the ecotype. So it is like different ethnicity, then different geographical area, different types of the regions in the globes can have different RFLPs. So they are used as a molecular markers. It's adapted to different environmental conditions. So other RFLP can be detected only by using specific cDNAs or the genomic clones. So these are RFLP, you can see it on a gel or after the southern blot, so it can be called a phenotypes. Because for the different ecotype, different ethnicity, we can have different types of the RFLPs. So next is the short tandem repeats. It is also a type of the molecular markers that will be discussed after knowing the other details of the RFLP. So classify the progeny of process as parental or the recombinant. Then RFLP will segregate as the codominant markers in the process. So restriction fragment, that is the product of the RFLPs after the restriction digestion. So homologous chromosome visible in the gel or detected on the autoradiogram. So we can directly see on the gel if it is ethidium bromide, or we can use the probe and we can see by the southern blot. So this one we already discussed earlier that we can prepare by 1992. There are almost 2000 RFLP were prepared on the 24 human chromosomes, we have the 46, 23, 22 pairs of autosomes and 1 pairs of the sex chromosomes. So by 1992, so many RFLP were prepared. Then the others are the genomic libraries or the cDNA libraries that is used for the physical mapping. Then STS, that is the sequence text sites, unique sequence in the genome, where we exactly know the chromosomal locations. Then ESTs, express sequence tag, these are the short STS that is identified with the cDNA libraries. So first we need to know what is the cDNA libraries. Then we know that sequence tag sites, and then the express sequence tag, which are the smaller or the shorter STS. These are all the molecular markers. Then single sequence length polymorphism obtained from the known genetic markers provide a link between the genetic and the physical map. Then this is the genetic map prepared by the recombinations or the pedigree analysis, and this is a specific chromosome for the bending patterns, which is a cytogenetic map. So genetic map provides the outline and the physical map provides the details because it is based on the sequence. So variable number tandem repeats, also called the minisatellites. Short tandem repeats, STRs, are the microsatellites. So variable numbers are the minisatellites, short tandem repeats are the microsatellites, and these are polymorphic. So number of the copies 2 to 5 nucleotide pair long, these VNTR and the STS. So one unit is 2 to 5 nucleotide, like the ACTG. This is in one, this is in the complementary. So it is a 2 nucleotide length. Then ST are defined in 2335 sites with an average distance of 1.6 centimorgans, or about 1.6 million bases between the adjacent markers. So we have almost 2335 sites of the STRs, and the distance between the two STRs is almost 1.6 centimorgans. Then RFLP mapping, the construct, it gives a detailed genetic map of the chromosomes. So we know the major molecular marker positions in the cytogenetic map. Because of that, we can make positional cloning feasible. Because of this, RFLP based molecular marking is possible. Then the genetic map that is prepared from the recombination mapping, supplemented with the physical map that is based on the sequence, that we can prepare by the restriction mappings and the sequencing. So large number of the genomic clones will be prepared that we will discuss in the form of the Contig. Then overlapping clones can be identified, and that is used to prepare a whole genome sequencing or exact location of exact sequence of a chromosome. So this is a positional cloning used to identify and clone genes with a known phenotypic effect in species. One is the short tandem repeats or the microsatellites. These are the repeats of the 1 to 13 nucleotides in length, linked to head to tail. In the previously it is 2 to 5, STS are 1 to 13 nucleotides. Unit number of the repeats are 5 to 10. So we have two things. One is the unit repeats, and the second one is the number of the total units. So fine scale mapping can give us 3 to the 4 allelic forms. So the two version of the STR here 1 to 3. So one unit is having 2 nucleotides, here 1 to 13, and it is between the 5 to 20. So here we have 1, 2, 3 repeats are there, and here 1, 2, 3, 4 repeats are there. So this is one band, this is 6 nucleotides. Okay, so it is smaller. This is 8 nucleotides, so it is larger. And this is present here. So like this, we made the positional cloning. This is chromosome 17, STR. This is the another example how to prepare the means mapping by the chromosome walking. So we can see here there is RFLP or it is a positions. Then we will prepare a restriction map of the clone. This is the gene of interest. Then this subclone, this area. So in this one, this is the step 1. This is the restriction mapping ESBBH. Then subclone BH, B to H, we will subclone this part. Then screen genomic library with the subclone as probe. Then we will screen it, we will sequence it. Then we prepare a restriction map of the new clone. The new clone starts from here to here. Then we subclone H and E. Similarly, we reach to the gene of interest in a stepwise manner. Cloning, sequencing, cloning, sequencing. Series of overlapping genomic clones allow researchers to walk from the RFLP into the gene of interest along the chromosome to the gene of interest. So we are walking by series of cloning and sequencing of the entire stretch of the DNA from the molecular marker position to the gene of interest. That's why it is called the chromosome walking. And when it is far from the positions of a molecular marker, that is a process by which sequencing is made faster, and that process is called the chromosome jumping. So each jump can cover a distance of 100 kb or the more. So the gene of interest can be identified or located from the position of a molecular marker such as the RFLP clone by the process of chromosome walking and the chromosome jumping. So this is the another way of preparing the overlapping sequence. This is called the contiguous genome sequencing or the Contig. The development of the vectors with a large insert capacity is able to product of Contig. Continuous overlapping clone fragment positions related to one another. Overlaps are created by partial digestions. So the DNA fragments, this is the cytogenetic map. From that, we have prepared the different fragments of the chromosomes and we clone them in the cosmid or the East artificial clone library, chromosome library, and we prepare a library. This consists of this entire area or the overlapping DNA sequences like this. So this is one Contig. Then we have prepared so many clones and each of them are overlapping. So that when we sequence them, we will align them and finally we'll find out the sequence of the whole Contig. Similarly, we have the overlapping Contig and we will find out the sequence of the entire area of our interest. So this is the clone Contig method. So this is the cytogenetic map or the genome map. Here these are the molecular marker positions. So between the A and B, it is almost 500 kb based on the restriction fragment length. This part is sequenced. This part is for fragment is prepared. Then the overlapping fragments are prepared. They were cloned, amplified, and this is a shotgun sequencing of the entire genome. So we have so many fragments, overlapping fragments. So clones are prepared, they were sequenced, and finally by aligning them, we can prepare a physical map. So this is the shotgun method or the clone Contig method. So in order to link the contigs, we need the in situ hybridization. So, or a probe generated from one end of a Contig in order to screen a different disconnected Contig. Probe production and identification is termed as walking, as we have discussed earlier. So we can connect the physical maps by the PCR-based sequence text sites. So PCR-based, these are the molecular marker positions, STS, are the molecular marker positions. So how it is done? Isolate the genomic cosmid clone. So we have the clone prepared. We subclone DNA into the M13 vectors. then we'll sequence it, identify the unique sequence, then we design the primers, then analyze the amplifications. So STS markers lie approximately 100,000 bases apart. These are the molecular markers like the RFLP, so that between the two STS we can prepare the different Contig and we sequence the entire area. These are detected by the Southern blotting, and library of the clones may be divided into pools, and each pool species are screened. So STS element may also be generated from the variable region of the genome to produce a polymorphic marker. Okay, so this is the chromosome. As we have seen, genomics is stratification chromosome library overlapping cosmate 5 to 10 kb, and this is by the primer extension we sequence this area. So this is the single and the double digestion by the enzymes, number of the fragments and their sizes. And as the lambda DNA is linear, the number of the restriction sites for each enzyme is, and like the restriction mapping we have done and we have sequenced it. So similarly, this is the as we have discussed earlier. So after the sequencing, the physical map is prepared. This is another example of the KPN1 sites. So all the KP1 sites fall in the 24.5 kb XBA fragment, as the 24 kb fragment is intact after XB1 XB double digestion. So the area between the two restriction endonucleases were sequenced. So this is the limiting condition. This is the fragment site. Each of the fragment is cloned. And this is the restriction mapping is prepared, and after the sequencing is done. So this is the conventional agar gel. We can prepare, we can get the only one set of the bands, but there is a technique called the orthogonal field alternative gel electrophoresis. So we have the positive, positive, negative, negative. So when the gel is running, so there is a change. Like the now it is this one is active, after that this one will be active, then in an alternative fashion, the both means the anode and the cathodes will be operational in a regular fashion in a series of reactions, so that the gel will move in a zigzag fashion, so that the resolutions will be better and we can get a better resolution of the number of the fragments. So it can be in a conventional gel electrophoresis, or it can be through the OFAGE or the orthogonal field alternative gel electrophoresis. So electrophoresis, electromotive force, they alternate between this orientation and this orientation, which is the 45-degree angle. It increases the resolving power. So similarly, we have the contour cloud homogeneous electric field, and we have the field inversion gel electrophoresis to increase the resolutions of the separation of the different bands. Then there is the transcript mapping by the hybridization between the genes and the RNA. In this process, the boundaries between the double and the single stranded region will mark the start and the end point of the mRNA. This is the expressed area of the DNA. So we hybridize the mRNA with the DNA. So this is the introns area that will form a loop. After that, it will be treated with the S1 nuclease. So it will digest the single stranded nucleic acid. So this loop are the single stranded, so they will be digested. So we'll only have those portions of the DNA who has the coding sequence, or they have prepared the mRNA. Then they will be treated with the alkali. So alkali will digest the mRNA. So we have only the DNAs. So from this DNA, we know what is the start and what is the end. So similarly, we know what are the exons in the DNA. So we know the precise start and the end points of the transcript in the DNA. and any of the introns it contains to be mapped onto the DNA sequence. So similarly, in the DNA, we know the sequence of the exons and the sequence of the introns. So this is the another way of doing it. So this is the 400 bases fragment. This is the gene, and this is the our clone construct. So we anneal the mRNA. So after that, it will be wherever sequence is preparing the or the express area, this one, they will align. So it is a double-stranded area. So the S1 nucleus will digest the single-stranded area. So only the hybridized area will be there. After that, the treatment of the alkali will digest the RNAs. So we have only the DNA. And in this way, we will know what is the start point for our transcription. This is the genetic process we can do for the animals and the plants. And in case of the human, we do the pedigree studies. For studies of the recombination frequency to prepare the genetic map. So here, this is the cytogenetic map, step 1. This is the complete sequencing that we have discussed earlier. By the shotgun method, we sequence the different contigs and we prepare the physical map. Then we have discussed by the hybridization, we can prepare the transcript mapping, exons and the introns, and we know the exact sequence. So this is the wild type allele of the gene. Then they are transformed into the vector. This is the mutant organisms, and we can restore the wild type. So in this way, we localize the physical map of the region and isolate it, and it can be sequenced. This is the Tetrad analysis for the preparation of the genetic map. This is the homologous chromosome. So this is one crossover. So when they are in the physical contact, it looks like a tetrad. That's what it is called, the tetrad analysis. So after that, we can see that this chromatid, the exchange of the segment of the chromatid. So these are the crossover and these are the non-crossover. This is non-crossover, this is non-crossover, this is crossover. Here, we can see this is a double crossover, this is a triple crossover, this is a quadruple crossover. And here there is no crossover. By the counting the chiasmata, we can know how many crossing over takes place. Chromosome maps by counting the number of the crossovers until the meiosis prophase. This is the counting the crossing over as a measure of the genetic distance. So this is a oogonia, there is 100 egg. In this 100 egg, we can see that non-crossover eggs are 70, single crossover are 20, double crossover are 8, triple crossovers are 2. So we can calculate the distance between the A and B as the 70 by 100, non-crossover 0, because it is non-crossover. So 1 into 20 by 100, 2 into 8 by 100, 3 into 2 by 100 equal to 0.42. So the distance between the gene A and B or the allele A and B is 42 centimorgans or recombination frequency 0.42. This is another example. Here it is a double crossover. So here this is the long wing gray body, vestigial wing black body, long wing gray body, vestigial wing black body. So there is a crossover, and after that we can see long wing gray body. This is the parental form. Vestigial wing black body is also parental form. So this is the parental, or the parental means. So there is no crossover. And here this is vestigial wing and the gray body. This is a mixed character. Long wing black body is a mixed character. So these are the recombinant ones. So the difference between the The distance between the two genes is 92 out of, this is 180 recombination out of 1000, it is 18%. Or that is 18 centimorgan between the two genes. So locating the approximate gene position in the human genome, this is by pedigree analysis. So here we can see AB, capital AB. So these are the recombinant one. This is small AB, capital AB, small AB. So here there is no recombinations. But in this case, this one is a recombination and this one is a recombination. So we can see the recombination frequency in this generation. So this is the three genes SC, EC, CV. SC the skewed bristol, EC the echinocytes, and CV the cross brainless wings. Okay, so this is the parental form. So all are having without plus signs, and here this is the hybridized one, the F1 generation. So one chromosome. After that, there is a crossing over between the wild varieties and with these characters. And in the second one, there is a crossover. So in the F2 generation, we can see these characters. So out of this, if you see Scott, echinus, cross veinless, this is the parental form, this is the wild type form. So these are the non-crossover products. Whereas the skewed and the echinus SC, these are the crossover products. So between the SC and the EC, we can see SCEC, SCEC, SCEC. So these are the frequency where there is a crossover takes place between the SC and EC. Similarly, between the EC and CV, you can see this is EC and CV, this is EC and CV. So these are the crossover product between the EC and the CV. So from this we can calculate the recombination frequency between the skewed bristles and the echinocytes, and between the echinocytes and the cross veinless wings, the genes among these. Okay, so a 3-point cross allows detection of the double crossovers, permitting to determine if the exchange in the adjacent regions are independent of each other. So we need to get the answer of the following questions, like does a crossover in the region between the SC and the EC that we have seen earlier occur independently of a crossover in the region between the EC and CV, the second gene, the region 2? This is the question 1. So whether there is interference of the second gene crossover with the first gene. Second question is, does one crossover inhibit the occurrence of the another nearby? This is the same questions in the different constant. Then we calculate the expected frequency of the double crossover based on the idea of the independence. So Breed and Olbreed's map the crossover frequency, the first one, SC and the EC, we have seen earlier. So 160, 130, 11, right? So here we see 160, 130. So this is between SC and EC, and this is the SC and EC. So this 1, 2, 3, 4. This one 1, 2, 3, 4. So it is 0.091. The region 2 is the other two, 190 to 148. Is 190 to 148 and 11. So the first region, the recombination frequency is 9.1%, and between the second region, the recombination frequency is 10.5%. So expected frequency of the double crossover is the first into the second. So it is 0.95% double frequency. But observed double frequency is 2/3248, only 0.0006%. Expected is 9.95, but observed is 0.06%. So there is a difference in the expected and the observed. So one crossover inhibits the occurrence of the another nearby. So region 1 crossover is inhibiting the crossing over the region 2, or the region 2 is inhibiting the crossing over of the region 1. So coefficient of coincidence C, which is the ratio of the observed frequency, that is the 0.0006, to the expected frequency, that is 0.0095, is 6.3%. coefficient of coincidence. So level of the interference is 1 minus C. It is 93.7% is the interference. So the interference is strong over map distance less than 20 centimolars. So if there are the 3 genes, then it is not only that there is a difference in the recombination frequency based on their distance, it is also possible that one crossing over is inhibiting the crossing over between the 2 other genes. That is the conclusion point. So in this way, we can place the positions of the various genes like SC and the EC we have calculated 9.1%. So it is 9.1 centimorgan. This is 10.5%, 10.5 centimorgan. Similarly, the other genes on the Drosophila are mapped like this. And these are the various techniques to determine the putative, means the genes, the function not known, okay, of the encoding sequence. Jew blotting. It is the evolutionary conservative of the DNA sequence that suggests functional significance. Homology searching. Gene database, searching to gene family related sequence. Identification of the CPG island. Region of the hypomethylated CPG frequently found 5' to genes in the vertebrate animals. Then identification of the open-ending frames, promoter of the splice sites. DNA sequence scan for consensus sequence by computers. Northern blot by RNA hybridization. Exon trapping method is the artificial RNA splicing assay for the exon identification. ESTs are the cDNA amplified by PCR that represent part of a gene. So these are the part of the cancer genome anatomy projects to prepare a physical map. So we have known the 3 types of the map. There is another way of the mapping in case of the bacteria is by interrupted conjugations. So in that case, through the conjugation tube, there is a rolling circle replication by which it starts from one specific site and it moves to another bacteria. So similarly, the mapping of the chromosome is done. And by this way, this is the position of the important genes and function of the gene cluster, the lambda genetic map. It is prepared like this. Thank you.